Good morning. We have Dr. Tulsiram from CSIR-NCL Pune, and his expertise is in natural products, their isolation, and studying their biosynthetic pathways. Now we'll ask him that how natural products actually owe importance to drug discovery and have societal benefits. So welcome, Dr. Tulsiram,
Thank you.
and we'll ask you to say, tell us and show to us how important natural products are for society.
As we all know, natural products are one of the major source of drugs in Indian society from the human society is formed. So what is required is we have not even 10 to 15% of the natural product sources have been explored towards their benefits, human benefits, but rest of this still untapped. So India is large potential to identify those molecules from those untapped sources and isolate, characterize them, and see their potential as a benefit in healthcare or agriculture or industry or in API as a pharmaceutical ingredients or intermediates. So one need to know, do systematic isolation and characterization and look for the new chemical entity from the natural products. Based on the folk medicine, Ayurveda, or um, samhitas, what we have, we can select the plant source and isolate systematically all the molecules and study their biosynthetic pathway and characterize them, what are the molecules or enzymes involved in their biosynthesis. To say one example, neem is a wonder tree in Indian
Context.
ancient
Yeah.
content. So it is known most of the major compounds have been isolated and characterized previously in 60s, 70s, and 80s. However, after that it is declined, and no biological study has been reported for any many neem liminoids, especially those are minor compounds as well as intermediates in the biosynthesis of final compound, which is azadirachtin A, known to be potent antifidant and anti-insecticidal activity. So what we totally involved in do tissue specific. First, we have to isolate the neem liminoids in systematic manner and characterize them and establish their identification in other sources. So using whether it is maybe LCMS or GCMS or HRMS equivalent methods. Once we identify them, then we'll look for the other plant sources in the same family or different family, so whether these molecules are present or intermediates are present. Then we'll isolated molecules will be sent to our collaborators either in CDRI
Yeah.
or across India, and you
Like
are
what
working
we are
that.
doing. Yeah.
Yeah. So then we will see that these molecules are mainly biological properties is not even in screened for what
True.
are their activity, especially in our case, bone regeneration. So epoxy is working very well there.
Yeah.
So likewise, we have started screening with different groups across India. And other side, we started working on biosynthetic pathway. What are the genes involved in the biosynthesis of
Yeah.
this such a complex molecule?
We go up to the level of molecular
Molecular,
level.
and these enzymes can be utilized for modification or for any in a synthetic modification in other molecules. So with this is a rich source. So we are involved systematic cloning and identification, functional characterization of individual genes involved in the isoprenoid or liminoid biosynthetic pathway. Likewise, one can start the work or look for the opportunity in natural products.
Yeah, and while we do this, I think you can always also tell us the importance that what are the limitations and what we have to keep in mind while we tap this knowledge from the plant products. For example, that we have to keep in mind that we do not destroy the we keep in mind the bioconservative part of it, and we have to keep in mind that we get it from the natural, from the renewable sources of the plant.
That's correct. That's
Yeah.
correct. Many of these natural products are very low level, present in very low level in their native source. So what one can do rather than keep on isolating and destroying the biodiversity, especially the endangered species, we can see that what are those molecules, whether we can get those molecules in alternative source, or whether one can systematically dissect the biosynthetic pathway,
Yeah.
so that those cascade biosynthetic
Yeah.
pathway cascade can be put it into genetically tractable heterologous system
Yeah.
to produce these
Large
molecules
amounts.
in a large amount as well as sustainable way.
Yeah.
That's
Yeah.
what we are trying to do in at NCL.
Yeah. Thank you so much,
Thank you. Thank
Dr.
you.
Tulsiram, that we'll be able to use your valuable knowledge
Thank you.
of plant products that you have. Thank you so much.
Thank you.